science,
engineering,
medicine,
chemistry,
physics,
biology, humanities, cardiology,
computing, public health, global
aeronautics, science communication.
Hello everyone, I'm Gareth Mitchell. Today, news from around the college, that's in just a moment, and also long COVID and how we're just waking up to the extent of its effect on those unfortunate enough to succumb.
These are people who used to cycle to work who can no longer cycle, people who now find it difficult to go up and down stairs. These are people who used to be full-time, full-on workers who now find it hard to turn up for a full-time job and have gone part-time. I don't know, and nobody can tell them what the long-term future holds for them.
But what do we know? An Imperial expert will tell us. Also in this edition, bioplastics, and should we put them in the brown recycling bin? And we have the story of an undergraduate project about one of Saturn's moons, and how the research has ended up in a peer-reviewed journal.
The Imperial College Podcast.
Well, let's jump straight in with some news. We have Justine Alford here. And Justine, just to start with, we're going to talk about the latest results from Imperial's COVID REACT study. Just a quick reminder for us, what is the REACT study?
Hi there. So the REACT study, which is led by Imperial College London, is an ongoing coronavirus surveillance study. And there are actually two arms to the project. There's the REACT 1 survey, which is swabbing hundreds of thousands of people across the country to look for the prevalence of coronavirus infection. So how many people are infected with the coronavirus at any one time, and where the biggest risk of infection lies across the country in terms of region and in terms of the people who are most likely to be infected. And then there is also the REACT 2 study, which is looking at the prevalence of past infection. It's doing this by checking people for antibodies to the coronavirus. So that's tracking how the virus has spread in the population.
So what are the latest results then? What are they telling us?
The study has actually released 2 reports over the last 2 weeks, so the team has been exceptionally busy. So the first one was the latest findings from the REACT 1 study, so the study on coronavirus prevalence. And the study found that the number of people testing for the coronavirus has continued to rise exponentially. And they actually found that infections have been doubling every 6 days. They've been documenting a huge rise in the number of people who are infected across the country. And in some groups, actually, compared to their last findings, infections had grown by 8-fold. And that was, for example, in London and in children who were in secondary school. They found that while the prevalence of infection was lower in people who'd been vaccinated, infections were still rising proportionally in this group of people who were vaccinated at the same rate as people who were unvaccinated. So there was a little bit of concern there and a reminder that people, are not completely immune to the virus when they've had their vaccine, but, just a reminder to get your vaccine as well to help protect you from illness.
Yeah, it was quite interesting that infections have risen even amongst vaccinated people. And these are people with both jabs, aren't they? They're double vaccinated people.
It was looking at people who were vaccinated in terms of single and double vaccinated. So there is, of course, a possibility that some people hadn't built up their full immunity at that point. And we do know that it takes a couple of weeks after you've had your second vaccine for you to develop that, strong protection against the coronavirus. So it is, of course, possible that some of the data from this study was coming from people who hadn't built up that full immunity yet.
Do we have anything from the REACT 2 study, the antibody study?
So this week, the REACT 2 study released a new batch of findings as well, and that was their biggest survey of antibodies yet. They looked at 207,000 people who had taken a finger prick blood test at home to check for coronavirus antibodies, and they found that compared to their last findings in January, that the number of people who had antibodies to the coronavirus had grown 4-fold, which really demonstrates the impact of the vaccination programme. But interestingly, they also looked at vaccination rates, which were very high. And they also looked at the antibody response in people who'd had a vaccine. And it was really encouraging in that they found that people who'd had both doses of a coronavirus vaccine, so either AstraZeneca or Pfizer, almost 100% of people who were double vaccinated produced antibodies against the coronavirus. And that's really important because we do think that high levels of antibody are needed to protect against infection, particularly of the Delta variant. However, what they also saw was that people who'd only had one vaccine, the antibody response was much more variable. And in some people, particularly older people, the prevalence of antibodies was below 50%. So this was really just a reminder that how important it is for everybody to get their second dose and to get it as soon as possible.
All right, Justine, thank you very much indeed. That's Justine Alford. Now, Joanna Wilson is also here. And of course, this week, the Olympics start, and then a few weeks thereafter, the Paralympics in Tokyo. And, well, Joanna, there's a fair bit of Imperial involvement, isn't there?
Absolutely, Gareth. That's right. So I'm really excited. We've got the Olympics coming up very shortly. So we've actually got one current student and one of our alumni who are taking part in this year's games. We're really excited about that to cheer them on. So firstly, we've got Daniel Roden, and he is a mechanical engineering student. He is a middle distance runner, and he is going to be taking part and representing Team GB in the 800 metres. And then we also have an alumni. So we've got Henry Fieldman, and he graduated from Imperial back in 2011 with a BSc in zoology. And while he was at Imperial, he rowed for Imperial's boat club, and he will now be taking part in the games, coxing the men's 8 in the rowing, which is very exciting.
And how about the Paralympics?
Yeah, so we have got lots of success previously in the Paralympics. So you may be aware of Dave Henson, who studied at Imperial. He is now with us in the Faculty of Engineering. He's won medals for us before. We've also had medals in sailing.
And on this very podcast, we have celebrated previous successes as they've happened. Just remind us then, because, well, there's a recent, excuse the pun, but track history here.
Absolutely. So if you cast your mind back to 2016, when we had the last Olympic Games, we had an Imperial alumnus, Mel Wilson, and a fellow member of our boat club, Zoe Lee, and they took silver in the women's 8 event. So really hoping we get some of the same success this year. And that's not all. We've had more recent success. But back in 1908, the Summer Olympics were actually held in White City, where Imperial now has a campus. So the White City Stadium was built for the very Olympic Games in 1908, and that was actually home to the finish line of the first modern marathon. We're all really excited to cheer on Henry and Daniel in this year's Games, and we're sure all of our listeners are looking forward to the Olympics too.
We certainly are. That's Joanna Wilson. Well, for many, Monday was so-called Freedom Day as the government lifted most of the remaining COVID restrictions. Meanwhile, many doctors are bracing themselves for what may be in store in the months and quite possibly years ahead. Long COVID, the chronic illness that follows in some people after the acute stage of COVID-19 has passed. It can have lasting, life-changing impacts on health and affect people's ability to resume normal life and even to work. For scientists, this is uncharted territory. One such researcher navigating into it is Professor Danny Altmann of Imperial's Department of Immunology and Inflammation. Ryan O'Hare caught up with Danny and started by asking, when patients present with long COVID, what kinds of symptoms do they usually describe?
They may have had quite mild infection or severe infection or even asymptomatic infection, and yet for several weeks or months or now years afterwards, many of them are describing the same kind of pattern of symptoms: fatigue, breathlessness, wheeze, chest pain, brain fog, a list of symptoms that goes on in different combinations through 40 or 50 symptoms that's out there in the world, and the many millions of people who've had COVID.
So do we know at the moment, can it affect anyone, or are certain people more at risk?
Again, there's an enormous data set that comes out of many heroic groups of sufferers around the world who've organised themselves into very large online groups. There's a sense from those data sets that females more than males, and there are immunological reasons why women might be more affected. Beyond that, I think it's hard to say. The only other point I'd add to that, somebody who's been extremely ill on intensive care for a long time with a viral load might, for example, have lung damage that you could see on a CT scan or an MRI. And we know that that's one subset of long COVID, but that's not the whole story. It's perhaps partly that, but it's also much more than that.
I guess in relation to that, do we know, obviously the picture's still emerging, but is there any evidence that there's a long-term risk associated with long COVID as well that may predispose you to other health conditions?
I think one of the reasons I worry so much for people with long COVID is the kind of uncharted aspect of it. At the moment, we have an awful lot of people reporting long COVID from the first wave, so in other words, well beyond a year. And in many cases, these are people who have a life-changing illness. These are people who used to cycle to work who can no longer cycle, people who now find it difficult to go up and down stairs. These are people who used to be full-time, full-on workers who now find it hard to turn up for a full-time job and have gone part-time. So this is the real thing, and I don't know, and nobody can tell them what the long-term future holds for them. So one of the reasons I became interested in this is that in our lab we have a lot of collaborations in Brazil, and one of the diseases we're especially interested in there is another totally different viral infection called Chikungunya, which is spread by mosquitoes. And the reason that we're so interested in that, in addition to our interest in related diseases like Zika or dengue, is that if you have chikungunya, there's a moderately large minority of infected people who will go on to get a kind of autoimmune arthritis that can stretch on for 3 or 4 or 5 years or more. And so it's the long-term after effect that devastates their lives, not the initial fever, because many of them lose their jobs or become suicidal. And that's the real burden to the Brazilian healthcare system. So you can see where I'm going with this in terms of the analogy, that's the thing that worries us.
Your team's just about to undertake some new research in this area, so with some funding from the NIHR. Can you tell us a little bit about what that will involve and what you're hoping to find out?
Yes, so what we really want to do is do something slightly new for our lab and really reach out to people in the UK who have long COVID symptoms, get them into the lab to give a blood sample, and really throw at that blood sample everything we know about analysing the immune response, looking at autoimmunity, looking at the array of antibodies to different proteins in the human body, and to see whether there are patterns of autoimmunity that correlate with those symptoms. We also want to look at RNA, look at the things that lymphocytes are making in those people, and see if programs of immunity are turned on differently in people with long COVID. One of the ways that I've tried to imagine this process when people have asked me about it is kind of by analogy to things like glandular fever, where if you know any teenager who's ever had glandular fever, it can take them years to feel normal again, and they become susceptible to minor infections for a long time. And I describe it as a bit like somebody throwing a hand grenade into your immune system. All the populations are completely topsy-turvy and unsettled for years. And that's a guess. But, we've got the technology to look at that in quite a lot of detail. And that's the third strand that we want to look at.
That's Danny Altmann speaking there to Ryan O'Hare. And you can see Ryan's written piece based on that interview. It's on our news website, and you can access that via imperial.ac.uk/news. And the full version of that audio is up there too. Well, with the barbecue season well and truly upon us, what are we meant to do with all that food packaging? For instance, what about so-called bioplastics? Can we put them in the brown recycling bin with our food waste? I must admit, I never really know. Lindsay Winton, Media and Outreach Officer at the Grantham Institute, has been putting those questions and many others about plastics and what to do about them to second-year PhD student Sarah Kakadellis of the Centre for Environmental Policy.
Globally, it's estimated that about 8 million tonnes of plastic leak into the ocean every single year. That's about the equivalent of one dustbin lorry every day being dumped in the sea globally. One of the reasons for that big number is because of the difficulty to recycle some of that plastic. It's often very contaminated. So especially for plastic packaging, which is what I research, a lot of it is used for food packaging applications, so you can imagine how sometimes difficult it can be to separate food waste components from plastic components, and so that makes it really difficult to recycle. It's also, I think, we tend to send our materials to locations where the system isn't necessarily in place to actually recycle it. So it looks good on our side, but then we just get rid of the problem, really.
There has been a proposed solution, which is bioplastics, and I wondered if you could explain what bioplastics are.
Broadly speaking, A plastic is considered a bioplastic if it is either bio-based, which means it's made of biological resources like potato starch or corn starch and increasingly waste materials. And a bioplastic can also be biodegradable. And again, biodegradable, a very controversial terminology, but biodegradable means that it can be decomposed by microbes under certain circumstances, so under certain conditions.
It's not as simple that all bioplastics can go in the brown bin by the sounds of it.
Yeah, exactly. Because of that, these two definitions, you can already see that not all bioplastics are actually biodegradable, even designed for biodegradability.
Do you think that one type of bioplastic should be prioritised for food packaging, or do we actually need both and they should be more clearly labelled?
There are many different types of bioplastics and just plastics in general, and that's because every material will have unique properties and be used in a specific application. So applications where you're likely to have food contamination, so if you think of salad packages, for example, in these situations, it can be quite useful to have biodegradable packaging that can then be processed and composted with food waste. For other applications, so if you think of a regular plastic water bottle, we have a really good system in place already to collect and recycle these materials. So substituting these with biodegradable plastics is not a good idea.
Do you think also there needs to be some sort of distinction in the labelling so it's really clear to people what is a bioplastic, what can go in their brown bin and what needs to go in the recycling? Do you think the labelling needs to be a little clearer?
Absolutely, and I think that's probably one of the key issues at the moment is the lack of consistent and clear labelling. So part of the research I'm doing now is also looking at the role of consumers and how we can support consumers in ensuring that they make the right decisions when they dispose of different types of plastics, but also to help the people who collect the waste and process that waste to actually understand which material they're dealing with. So yes, clearer labelling, less space for greenwashing, which will also empower the consumers to act.
Finally, as individuals, what can we do to reduce our plastic consumption? Do you have any tips?
One of the best ways, I think, is to think more about reuse models, whether that is to bring your own Tupperware to work or places where you can also bring your own coffee cup. So also places like bulk buy stores, so where you can buy in bulk as opposed to in smaller individual packaging. I think we also shouldn't think just of packaging when we think of plastic, which I think is very common. Another area could look at clothing. One of the best ways to reduce our consumption of plastic is actually to stop continuously buying new clothes. And finally, I should note as well that the majority of plastics are still fossil-based, so they're tied in with the fossil fuel industry. And so by reducing our reliance on fossil fuels, we're also sending a signal to the petrochemical industry, and that in turn, hopefully in the next years, will reduce the production of plastics in the first place.
That's Sarah Kakadellis speaking there to Lindsay Winton. Well, finally, it was one of the most spellbinding discoveries of the Cassini mission to Saturn. From the south pole of Enceladus, one of the moons of Saturn, plumes of water and ice particles jetting out like geysers into space. Ever since, the jets have been the subject of intense scientific scrutiny, and some of the latest such research comes from Imperial, and guess what? It's from an undergraduate project that turned into a peer-reviewed study. Hayley Dunning has the story, and it begins with the young researchers introducing themselves.
I'm Ravi Desai. I'm a postdoctoral research associate in the Space and Atmospheric Physics group at Imperial College.
So my name is Sochi, so I'm just graduated from Imperial College Physics Department.
Hi, I'm Sunil. I just finished my last year of MSci Physics.
Tell me about the research. So what did you investigate and how?
So the project we worked on was studying molecules discovered in Enceladus's plumes. So Enceladus is one of the moons of Saturn. It was discovered to have a large ocean of liquid water inside it, and this water was found shooting out of the South Pole in jets. And this project was focusing on how long molecules inside this plume could survive for?
Yeah, so one of the main ways that the molecules decompose at this plume is based on the emission of the photons from the sun. They dissociate the molecules through a process that's called photodecomposition. We use some cross-section, so basically the probability of the photon dissociates the molecule, which some are determined experimentally and some are determined through some theoretical calculations to predict, based on the flux of the photons as well as the concentration of the molecules, how long they will survive outside the plume.
We use computer to simulate their lifetimes, so we could have an insight of how long different species of particles could last at different places in the solar system.
So why did you pick this topic? Why is it important to know that?
It's very important to understand the composition of the plume of Enceladus, because we want to understand what the composition of the ocean is inside Enceladus. And if we can understand that environment, we can maybe even understand if life is possible in that environment. But Zecchi and Zinni didn't only calculate the lifetime of molecules in the plume. They went away and they came back the next week and they'd actually calculated the lifetimes of pretty much every stable negative ion they could find at every single location in the solar system, which was quite remarkable and led to some really interesting results.
I think another reason that we chose this project when we were choosing from like hundreds of projects when we were in first year is because it would be the first time in the college that we have the chance to get our hands on real data collected by Cassini spacecraft. That would be something that we have never done when we were in first year.
It's impressive stuff. So is that one of the reasons you decided to get it published? Whose idea was it?
I think that was my idea. When they calculated all these rates, it was immediately clear that this hadn't been done before. And what's novel about this is that people who study the Enceladus plume typically work within planetary science, they work within space physics, but some of the calculations which Zecchi and Zinni did, it comes from fields of fundamental chemistry or astrochemistry. So they combined information from two different fields to get their results, which is why we decided to write them up.
I didn't believe Ravi when he first said, well, this is publishable results. After all, it's a first-year project. But as the project went on and we went through more references and done more calculations, and we find, probably there is something novel about this project.
I was going to ask those, Zecchi and Zinni, what was it like then working on a peer-reviewed paper so early in your careers?
It feels quite unrealistic because we have been reading and referencing lots of paper, but it is first time that there's going to be an actual paper with our name on it instead of like lab report. It could be a small thing, but it still feels very different. It feels even better when we think about what we have done could help more and more researchers in the future.
And how did it feel when you finally got the acceptance that this was going to be a published paper?
Because it's been a long time from when we first finished the first year project, which is like about 3 years ago, because we are already graduating from the fourth year. It's sort of almost like a reminder that we did something that is as impactful as that. It's sort of like a time for us to reflect on the degree as a whole from the first year to the fourth year, how much progress we have made and how much improvement will have been made. When you're considering whether you're going to apply for a PhD or going to do research eventually, I think the undergraduate courses are a very great testing ground for it. It's not like before we go into Imperial College, we know we're going to be a researcher one day. No, because we are not sure. And that's why we tested our abilities and tested our interests and sort of connect with the research community to see, are we really suitable for research one day in the future?
Now you are both about to graduate, so what's next for you?
As for me, I have to say I'm not very sure at the moment. I might be doing some data analysis in other fields, but I will always apply what I have learned from this project.
For me, obviously it's all planning at this stage, but I'm planning to take a gap year first, learn some other skills other than physics to sort of take a break before the PhD that I'm going to apply for, I think, next year. My career plan for now is, I think similar to Ravi's, is going to research and I guess advance the scientific frontier by a little bit.
Oh, I love that. Advancing the scientific frontier. A little bit. And whether you're an undergraduate or a top professor, that's science, isn't it, really? Advancing one little bit at a time. That report from Hayley Dunning, bringing this edition of the podcast to a close. The customary reminders as to where you can find us. Basically, we're on all your favourite podcasting platforms. And if we're not, let us know if there's one that you want to find us on. But we're on iTunes, Spotify, YouTube, and SoundCloud, and Entail Visual Podcasting. Amazing. All right, well, I'm Gareth Mitchell, bidding you a fond farewell for this month. I'll be back with more next time. In the meantime, from all of us here on the podcast team, thanks for listening, and goodbye.